Hello, everyone. This is my first time coming to Gatsby Summer School, which I'm sure is its correct name. So I just want to briefly introduce myself, and then we're going to talk about something kind of insane that I did. So this is my curriculum vitae. So I did my undergrad at Imperial. If you're at Imperial, I might still be on every TV in the building because that's kind of my curse. Then I did my PhD at Edinburgh. If you know, you know, is the Imperial thing. Then I did my postdoc at the Max Planck Institute for Molecular Plant Physiology in Germany. And then I came back to the University of Edinburgh, where I'm doing this strange hybrid role of being a postdoctoral researcher / university teacher. They are emphatically not saying it's lecturer. One who lectures is whatever. So my general interests are development. So this here is an Arabidopsis root meristem, and this is where kind of all the magic happens with root development. And over here is a diagram I found that illustrates cell-to-cell communication. So we have molecules being produced in one cell, and they move to neighboring cells through these pores in the cell wall called plasmodesmata. Right? And so what I want to talk about today is where they fuse together. So essentially, I had this really stupid idea where I wanted to kind of investigate molecules that move between cells in plants using a different approach. Now, in mammalian biology, they have this really cool method where they can build little organoids. So what that means is they can build a very tiny little brain, essentially, made up of all the relevant neurons, and it functions like a tiny brain, and you can make hundreds of them and do loads of experiments on all of them. That technology does not exist in plants. So we want to try and figure out how to make that work. So essentially, the goal would be you have multiple cell types, you stick them together somehow, and then you study what moves between them through the plasmodesmata that form in between the cells. And so you basically have to, for this to be a functioning tissue, you need it to do three things. One, they have to stick together. They have to actually be one tissue. Two, they have to do some kind of cell-to-cell communication. Otherwise, they're just tissues next to each other. And number three, you have to actually physically be able to do it, which by far turned out to be the hardest part. So this is a Callus culture, and we figured this was a pretty good place to start. So if you take plant tissue and you expose it to the hormones auxin and cytokinin, what you get over time is the proliferation of these cells like this. And if you extract them and continue culturing them on media containing auxin and cytokinin, they'll just keep proliferating. And they can just keep going and keep going. You cut them into smaller bits, you keep it going, and it can go for decades in continuous culture. So we figured this would be a pretty good place to start. So what we did was we tried to glue together a bunch of callus to see if anything would stick together and if the cells would exchange molecules between them if you just stick them close together enough. And it turns out, without much effort, they just do it. So the goal was this. You take a group of individual calli. So you have, all around the outside, you have the green ones. So these are expressing GFP, right? Just GFP on its own. And in the middle, you have a callus that's expressing a nuclear localized red fluorescent protein, right? So you need this so you can tell the difference between the two things, because after a while, they look really similar. And the goal is essentially this. You have two groups of cells. You have what we're calling the producer cells. So these are our cells that are producing GFP. So GFP on its own just loves to diffuse. It will move between cells. It will wash all over the place if you let it. Nuclear localized fluorescent proteins don't. They will stay in the nucleus. So therefore, if we are able to successfully stick these two things together, we can watch as green moves from one to the other. And so when we find a cell that contains a red nucleus and suddenly a load of GFP, that GFP can only have come from one place. And that has to be a cell that it's stuck to. You with me so far? Right, half the people nodding, okay. Yeah, make some noise, people. Right, so what we find is if you look at it really, really up close, you can see something that looks like this. So this is a 3-panel image. So this here is the YFP signal. So YFP, GFP, we use them interchangeably in this because they move the same. And you can see here these cells highlighted with the arrows, in particular the nuclei, also have red nuclei. So in these particular cells, the GFP has managed to move from the GFP-only cells into the nuclear-localized RFP cells. So in this particular Callus graft, we were able to achieve some cell adhesion and GFP moving from one to the other. But to test this properly, what we had to do was some controls. And the funniest part of this is, look at the N number for both of these and realize that a master student had to do with analyze every single one. So we took GFP that had been tagged. So it's localized to an organelle and it can't just diffuse between things. So when we use an ER localized GFP, we never see it. In 49 repeats, we didn't see it once in any cell that had a red nucleus. And when we used tubulin associated GFP, look at that number. Each one of them takes like half an hour to image. We don't see it either. So that gives us pretty good confidence that this really is GFP moving from one cell to another type. So we managed to just kind of clumsily stick together a bunch of cells and watch them exchange macromolecules. That was easy. So this image, I hate this image. So this image represents 3 straight months of my life, right? So basically, I did the same Callus graft. I took 2 different kinds of Callus, stuck them together, let them grow, and imaged them. So this time what I use is 2 different plasmodesmata markers, right? So MP17 GFP marks plasmodesmata with a green fluorescent protein. PDLP5 cherry marks plasmodesmata with a red fluorescent protein. Therefore, if you stick them together and you can successfully find the exact point at which they have connected, you should be able to image a complete graft junction with green on one side and red on the other to show that 2 different cell types have successfully formed a joint. This took me a long time to find this exact spot, but there they are. This is the point, and there's the red underneath and the green on top. So they are able to join together and form plasmodesmata and exchange molecules. So what we really needed to try and figure out to be able to do anything cool with this is, can we make this smaller? So at the moment, this kind of region in purple is the only bit that's in contact with its neighbor. So that's the only bit that has a chance of forming a kind of connection or a cell adhesion or any macromolecule exchange at all, right? And that's kind of a lot of wasted tissue. So what we needed was to try and make this smaller, get more cells mixed all together to try and make a completely joined up tissue. So we had to figure out a different starting material. And so what we went with was a suspension culture. So, oh wow, that looked much higher resolution on Google. So I don't know how well you can see this. A suspension culture essentially works by, you take some callus, this constantly proliferating tissue we have, and it happens to be very loose. It's called friable if it's good, right? So it just breaks apart. And you put it in a shaker containing growth medium, and you let it shake, and bits of it gradually flake off, and as they flake off, they continue to grow. So you get these small clumps of sort of somewhere between 2 and 4 cells, and then the motion breaks them again if they get too big, and you end up with this constant suspension culture of little cells. So this would be really perfect, because we could just mix these together, spread them out, and see if they grow together in a kind of tightly knit lump. Problem with that is, they take a really long time to establish. It takes months of them swirling and swirling and swirling to try and break apart and be able to generate enough of it. So what we decided is we would try to think of a really fast way to make a suspension culture. So the master student comes to me, and we're thinking about this, and we go, what if we could just agitate them more or faster? And then she says, what if we put them in the tissue-lyzer? And I said, does anyone know what a tissue-lyzer is? That's a tissue-lyzer. Does anyone know what this does? Yes, right there. It's essentially like a paint mixer. That goes, and plant tissue goes in, powder comes out. So now you can see why when she comes and says, let's put it in the tissue-lyzer, and I go, that's a normal reaction. And then she says, why not? And I said, huh. So we put it in the tissue-lyzer, and we put it on low, and we didn't use the metal balls, and we didn't freeze anything. And it turns out what you get is just a kind of paste of cells all down the side of the tube. And if you take these cells by scraping them out of the inside of the tube, and you mix them together, and you kind of sort them into small and large, what you're able to get is a much smaller, much more mixed organoid. So these are still pretty crude because we have very little control over it. But you see here, using large pieces, we have sort of large piece stuck together. If you go really small, they're really mixed up. And you can see here that basically everything that has a red nucleus, or almost everything that has a red nucleus, has GFP moving through it. So they're really tightly stuck together and mixed up. And to put this on a kind of scale, I've arranged them so that the scale bars are approximately the same size in each one. So we've managed to make this a lot smaller and a lot more dense and a lot more stuck together. And then we thought, what if we just tried to regenerate this into a plant? Would it end up as a kind of a plant that's got basically two different kinds of tissue making up one thing? Would we be able to make a whole chimeric plant? Because that would really open up the ability to study cells, molecules moving between different things in different tissues in a plant, because you could just have clonally different things. You could have a mutant layer and a wild-type layer, or backwards, or anything like that. And so what we did is we took two different accessions of Arabidopsis, so Columbia and Landsberg erector, and they're convenient because they are different enough that you can tell the origin of different RNAs. So if one of them moves into the other one, you can figure that out by PCR or sequencing. And what we were able to do is actually able to get some of these to kind of regenerate. Did they live very long? No. But we're still working on that. And you get something that looks kind of like this. This is the hypocotyl of a regenerating plant from a small mixed up callus. And what you can see is you kind of have layers of sort of just GFP. You have layers where the red is present. You can see that the green moves into one to the other. So you can kind of make this chimeric plant, which we're hoping, when we finally get really good at this, can be a really cool new platform for doing really, really cool molecular biology and synthetic biology and all kinds of fun stuff. So this is nominally supposed to be a careers talk, so I wanted to end with some conclusions. So in plant science, you get to do three things, really. You get to consider interesting questions, like, what insane thing can I do today? 2, you get to work with brilliant people who go, hey, what if we just put this plant on a tissue-lyzer? And 3, you get to do ridiculous things. So you get to have creative fun. All right, everyone, that's the end of my talk.